Thanks for tuning in. This video will showcase our recent preprint showing correlations between the phytochemical tolerance of parasites and the chemical ecology of their insect hosts. Feel free to check out the preprint. Plants are a source of many antimicrobial substances that protect these sedentary organisms from exploitation by microbes. These same substances can provide a barrier to infection in plant-consuming animals, as evidenced by the use of plants such as cinchona, garlic, and thyme for medicinal purposes. Plant compounds can also affect resistance to infection in plant-eating wild animals, from monkeys to monarch butterflies. If plant compounds in host diets shape their associations with infectious microbes, then the chemical tolerance of gut parasites should reflect their exposure to chemicals that are commonly encountered in the host gut. This sounds plausible, but how could we test such an idea? The trypanosomatid parasites of insects provide an ideal system for comparing the phytochemical tolerance of related parasites that infect hosts with differing diets and gut chemistry. This family of flagellated protozoa includes species associated with phloem-sucking and seed-eating bugs, nectar and pollen-consuming bees, and blood-sucking flies, such as mosquitoes and sandflies. Outside of entomology departments, the most well-known trypanosomatids are the insect-vectored species that infect humans, causing some of the world's most devastating parasitic diseases, including leishmaniasis, which is second only to malaria in its global burden of human morbidity. Hundreds of plant extracts and their chemical components have been tested against these parasites in the search for new, affordable therapies. Plant chemistry has also been linked to resistance of plants to their own trypanosomatids. These are the phytomonads, which can be devastating for some crops in tropical areas, such as palms. The extensive interest in clinically and agriculturally important trypanosomatids provides a base of knowledge against which to compare phytochemical tolerance of lesser-known insect-restricted species. The nectar and pollen-based diets of bees provide an excellent opportunity to study how a unique diet and gut environment has shaped parasite communities. Nectar and pollen are extremely rich in flavonoids that have strong effects against Leishmania species. Honeybee gut bacteria and the genomes of bees themselves exhibit adaptations to high flavonoid exposure. So, has nectar and pollen also shaped the trypanosomatid parasite communities of bees to favor strains and species with high flavonoid tolerance? We used experiments on 2 honeybee gut trypanosomatids that are closely related to the human-parasitic Leishmania to address this question. Intriguingly, only 2 trypanosomatid species have been characterized from honeybees, perhaps reflecting the difficulty of colonizing their flavonoid-rich gut environments. They are Crithidium malefici, found in ailing honeybee colonies in Australia in the 1960s, and the more recently described Lotmaria passim, passim meaning everywhere, which has emerged as the dominant honeybee trypanosomatid worldwide since its discovery right here at the Bee Research Lab by Ryan Schwarz and Jay Evans. These parasites have been found in over 80% of colonies in some national surveys, including in the US, and associated with colony collapse on 3 continents. Related parasites in bumblebees have a range of chronic effects. They include slowed colony growth, impaired foraging efficiency, and reduced overwinter survival. As a result, beyond their fundamental importance in understanding host-parasite associations, these trypanosomatids are of some concern in managed honeybees, which could benefit from phytochemical-based therapeutic treatments. We compared these honeybee parasites to the related mosquito parasite, Pythidia fasciculata, which infects adult and larval mosquitoes. Thanks to John Filosa for this great video, and Megan and Michael Popolonis for generously providing their isolate of this species. Now, although adult mosquitoes do feed on nectar, they rely on phytochemically deficient blood rather than pollen as a nitrogen source in the adult stage. On the other hand, larvae are exposed to considerable levels of phytochemicals in the shallow, debris-filled ponds in which mosquitoes breed. These larvae filter feed on bacteria and particles in the water column, and sometimes even browse decaying plant material directly. Compounds derived from wood and wood decomposition can influence larval survival and could shape mosquito parasite communities as well. We predicted that parasites of bees would have greater tolerance of flavonoids found at high concentrations in pollen, whereas parasites of mosquitoes would be more tolerant of compounds derived from wood decomposition found in larval habitats. To test these predictions, while also evaluating potential treatments for managed bees, we measured growth rates of two honeybee parasites, Pythidium mellifici and Lotmaria passim, and two strains of the mosquito trypanosomatid Pythidia fasciculata in the presence of 26 chemicals or plant extracts, then compared the inhibitory concentrations, a measure of phytochemical tolerance, across the two parasite groups. Of the compounds tested, 11 had quantifiable inhibitory effects on bee and mosquito parasites. Some of these, such as thymol, have been recorded in floral nectar at concentrations comparable to those needed for parasite inhibition, between 20 and 30 micrograms per milliliter. That suggests the potential for antiparasitic floral resources to mitigate bee infection. Note that the thymol LD50 for honeybees is an order of magnitude higher than the concentrations needed for parasite inhibition, suggesting that this compound could also be useful for treatment of infection in managed colonies, where thymol is already used as a fumigant to eliminate parasitic mites. Also noteworthy is that the inhibitory concentrations for these insect-restricted parasites were within the range previously found for sandfly-vectored Leishmania species, as indicated by these horizontal lines, suggesting that these plants' nectars could mitigate Leishmania transmission as well. The most striking difference between the bee and mosquito parasites was intolerance of the flavonoid, chrysin, which inhibited growth of both strains of mosquito parasites at less than 15 micrograms per milliliter, but had minimal effects on either bee parasite up to 50 micrograms per milliliter. In other words, tolerance to this compound was at least 4 times higher in parasites of bees. Flavonoids, including chrysin, are found at high concentrations in pollen and even higher concentrations in propolis, which bees gather and use for colony construction and repair. The high tolerance of bee parasites to flavonoids is consistent with the bees' own adaptations to high flavonoid exposure, demanded by their pollen-rich diets and use of propolis. It would be of interest to know the mechanisms underlying this high tolerance, given the interest in use of flavonoids as a treatment for Leishmania and the potential for subsequent emergence of drug-resistant parasites. The mosquito parasite, on the other hand, was more tolerant of 2 compounds associated with wood decomposition, the benzenoid vanillin and the phenylpropanoid cinnamic acid. Both of these compounds result from degradation of lignin, the main building block of woody plants, and can reach high concentrations in decaying woody debris-rich larval ponds, again suggesting a role for host diet-associated chemistry to shape the ecology and evolution of gut parasites. Finally, the majority of the compounds tested, although they were chosen for their activity against Leishmania, had weak effects on these insect-restricted parasites. This was particularly notable for most of the flavonoids, which are present in nectar consumed by both bees and mosquitoes. However, the sandflies that vector Leishmania also consume nectar, raising the question of why flavonoid tolerance appears to have lapsed in this two-host parasite. One explanation could be the loss of an antioxidant protein, catalase, in Leishmania. Now, antioxidants are likely important in buffering the oxidative damage that these compounds can cause. And interestingly, all of the mammal-infecting trypanosomatid lineages lack catalase, which appears to interfere with infectivity in mammals, suggesting trade-offs between phytochemical tolerance and mammalian infection. Summarizing the results, bee parasites had higher tolerance of a pollen flavonoid, corresponding to the flavonoid-rich diets and hive chemistry of honeybees. On the other hand, mosquito parasites were more tolerant of compounds associated with decaying woody debris found in larval habitats, an important stage for parasite transmission and establishment of infection. And overall, the insect-restricted parasites had higher tolerance to oxidative stress-inducing flavonoids than did the mammal-infecting Leishmania, suggesting trade-offs between tolerance of phytochemicals and the ability to infect mammals. This could enhance the potential for phytochemical-based treatments of these neglected tropical diseases. So why do these results matter? Most immediately, they suggest floral resources and therapeutic treatments that could be used for management of infections in honeybees, and possibly other bees vulnerable to spillover infections from managed colonies. In terms of parasite ecology and evolution, they provide suggestive evidence for the role of plant compounds in host diets as a driver of host parasite specificity, which could contribute to the still unexplained patterns of trypanosomatid infection found in insect communities. Finally, from the perspective of evolutionary medicine, they underline the complementarity of research on bee trypanosomatids and insect-vectored parasites of humans, where Leishmania research can suggest compounds for treatment of bee diseases, and bee parasites could provide resistant models for understanding the basis of tolerance of inhibitory compounds. This knowledge will be important for developing and preventing resistance to plant-based therapies for clinical infections. But wait, these supposedly insect-restricted parasites may be more than just lab rat surrogates for human infection. Forthidia fasciculata has long been considered a benign model for study of trypanosomatid physiology. But a strain of Forthidia fasciculata, which is more closely related to several bee trypanosomatids than to Leishmania, was recently implicated in an outbreak of visceral leishmaniasis in like illnesses in Brazil. Visceral leishmaniasis is not something to be trifled with. It is almost invariably fatal if untreated and affects mostly children in the developing world. And bee parasites may likewise turn out to be more than just models of parasite spillover and host shifts. Pythidium malefici was recently found in blood samples from bats, lemurs, marmosets, coati, and other wild mammals in Brazil, indicating its viability in mammals, including primates. Research on the phytochemical tolerance of bee-associated trypanosomatids can therefore provide insights into not just honeybee management, wild bee conservation, insect parasite ecology, and the evolution of chemical resistance among flagellates, but also the treatment of emerging insect-vectored zoonotic diseases. To sum up a punchy poem, diverse flagellates in insects writhe, yet in honeybees few remain blithe. Match of parasite host, unexplained still for most, could be shaped by plant compounds, sharp scythe. A grateful thank you to my colleagues and sponsors who have supported, enabled, and improved this work. I hope you enjoyed the video. Feel free to check out our preprint, drop a note, or leave a comment in the chat.